Welcome everyone to this webinar on traceability in nuclear medicine dosimetry. My name is Ana Denis-Bacelar, and I am a principal scientist at the National Physical Laboratory, which is the National Measurement Institute in the UK. I have nothing to disclose. So let's start with some definitions. Metrology is the scientific study of measurements, and its main activities include the definition and the realization of systems of units which are internationally accepted, as well as establishing traceability chains by disseminating the importance of accuracy in measurements and therefore the need to measure the associated uncertainties. So metrology provides a measurement infrastructure which is stable over time, comparable between different locations, as well as coherent, so that the results from different methods and users can be brought together. This is very important for medicine and the field of nuclear medicine dosimetry, as we want to ensure that all the measurements are accurate and reproducible between different hospitals and countries, even when we're using different dosimetry workflows. This is of vital importance to ensure the safety and efficacy of using radiopharmaceuticals, as well as to enable comparisons between results from different multicenter studies. One of the key concepts in metrology is metrological traceability, which is defined as the property of a measurement whereby the result can be related to a reference, usually a primary standard, through a documented unbroken chain of calibrations, each contributing to the measurement uncertainty. At the top of the traceability pyramid, we have the SI International System of Units. And to bring these units into reality, we must realize them through a hierarchy of measurement standards. First, we have the primary standards at the top, which are held at national measurement institutes across the world, like us, NPL. These primary standards are then used to calibrate other standards all the way down into measurements by the end users. So, for example, it could be hospitals using radionuclide calibrators for activity measurements prior to administration to the patient. And as you might expect, at each step that we go down this traceability chain, the measurement uncertainties increase, and the longer the chain, the higher the uncertainties. The BIPM is the International Bureau for Weights and Measures and oversees the validation of these standards across institutions to ensure there is equivalence in measurement capability. And we will see later a good example on why we actually really need this equivalence. So for nuclear medicine dosimetry, there are two main units which are of relevance. Firstly, we have the gray as the unit for absorbed dose, but we also use the becquerel, which is the unit for radioactivity. As the radiation for nuclear medicine dosimetry it depends on the individual uptake distribution of the radiopharmaceuticals, direct traceability to the gray is not possible like it is the case in external beam radiotherapy. So the main unit in terms of achieving traceability in nuclear medicine is the becquerel. And this is what a very simplified view of the traceability chain looks like for nuclear medicine dosimetry, starting from the development and the dissemination of the relevant activity standards for each radionuclide of interest into the patient administration and through all the different steps, including calibrations, patient imaging, image corrections, image reconstruction, delineation, quantification of activity, time activity curves, dosimetry, and even radiobiology calculations. Usually the traceability stops at the point of administration, and in many cases, traceability of the administered activity is also not guaranteed. To have traceability all the way into the absorbed dose calculation, it is important we understand each of these measurements and their associated uncertainties. So let's look at the first step in the chain, which includes the realization of the becquerel through a primary standard. This is a standard which is established by using a primary reference measurement procedure, like is the case for radionuclides, or created as an artifact chosen by convention, like in the case of the old definition of the kilogram. It is an absolute measure that does not rely on any other type of calibration. Bringing this concept back to nuclear medicine, for radionuclides, we develop primary standards for radioactivity, where we have a standard for each radionuclide, which give a measure of activity concentration. There are different primary methods that can be used, and the choice depends on the decay scheme of the radionuclide, but they all have in common that they only use the nuclear decay data and the experimental arrangement of the instrumentation to detect the radioactivity to achieve the absolute counting efficiency that is needed to have a primary measurement. Here we can see what the primary standard for yttrium 90 looks like, with measurements from multiple international metrology labs. And as mentioned earlier, the aim is to ensure equivalence between these national standards. So in the Y scale, you can see the so-called degrees of equivalence, which represent the degree to which the value of a measurement standard is consistent with the key comparison reference value. You can see that deviations from 0 for all the institutions are well below its uncertainties. And this is a very good thing, and it gives confidence that these labs have a good primary standard. You can actually find other key comparison and measurement capabilities of different metrology labs within the BAPM website. However, primary measurements are very complex and time-consuming, and therefore are used to calibrate secondary standards, which are also held at national measurement institutes. These are much simpler setups which enable a faster and more straightforward measurement that can be used to disseminate the primary standard further down the chain to the end users. So the idea here is that you take two samples from the same stock solution. One you use it for absolute counting, which gives you the activity concentration, and the other one is measured in an ionization chamber, which provides a measure of the current in units of picoamperes per gram. And putting these two together, we get a calibration factor for the secondary standard instrument, which of course is only valid for the measurement geometry, for that measurement geometry and that radionuclide. As we saw in the traceability pyramid, these secondary standards can then be used for calibrating other instruments and to provide calibration services. So let's look at an example of the development and dissemination of these activity measurements and why the concept of international equivalence, which I mentioned earlier, is very important. Here we have radium 223, which is still the first and only alpha meter with marketing authorization, following a statistically significant overall survival improvement in patients with metastatic prostate cancer. At the time of development, there were no primary standards available, so the company Bayer worked with a single national measurement institute for its development. And the trial took place, and then the results of the trial were published in 2013. And 2 years later, in 2015, a second metrology lab started developing their own standards for radium 223 and was repeating the measurements, found that there was a problem with the original standard from the other lab, which led to a 9% bias, which subsequently was also confirmed by the original lab. So in fact, patients had been receiving 55 kilobecquerels per kilogram instead of 50, which led to the company needing to change the product label to reflect this error. This was a very good example on why we need to validate our standards internationally before starting clinical trials. And if this was external beam radiotherapy, a 9% error on prescribed dose will not be tolerable. So for emerging radiopharmaceuticals, we must do better and ensure that these measurements are available before wide use in trials. Very important for novel radionuclides and other alpha emitters still. So I have mentioned that the need to calculate measurement uncertainties to ensure traceability, and this is perhaps one of those things that gets overseen as not being important. So let's dig into this a bit more. Uncertainty is a parameter that characterizes the dispersion of quantity values attributed to a measurement. It's a very helpful parameter that can help assess the reliability and the fitness of a measurement result. It gives confidence that the limitations of the measurement process are understood, and it also facilitates the improvement of measurements. We cannot make meaningful comparisons between measurements without uncertainties. So let's look at a very simple example. Let's say a patient is treated with any given therapy, I don't know, let's say lutetium dotatate, and you have access to SUV PET baseline data prior to the treatment, plus 2 follow-up scans. If you see these SUV values, will you say that the patient has responded to the treatment? I guess probably most people will say yes. Now let's add some uncertainties to the SUV values. Would you say the patient has responded now? Well, yes, uncertainties are reasonably low and the differences are statistically significant. Now, the uncertainties are much larger and therefore we cannot really say with any degree of confidence that the patient has responded. So I hope that you see that the uncertainties are useful and needed to inform any decision making. But actually, how do we calculate uncertainties? There is, of course, the GAM, or the Guide to Uncertainty Measurement document, which is available in the BIPM website and is like the Bible of metrology, together with the International Vocabulary of Metrology, which is also a very useful resource to check on simple definitions of different metrology terminology. But of course, these documents are very general for any type of measurements and not just for nuclear medicine dosimetry. So for more specific guidance on uncertainties, you can follow the ENM guidance on uncertainty analysis for molecular radiotherapy absolute dose calculations. These guidelines propose a method to evaluate uncertainties on a typical clinical dosimetry workflow, calculating mean absolute doses to organs and tumor levels, to the organ and tumor levels, and includes uncertainties for different steps in the chain. For example, it brings together the calibration data used to convert from counts into activity, together with calculating and fitting recovery coefficient data to then determine the recovery for the desired volume of interest, the actual calculation of the volumes or masses to then calculate the dose factor, which is determined from fitting a volume-dependent function to a sphere dose factors data. And then we can bring all of this together to quantify the activity in the given volume of interest, fit a time activity curve, and the guidelines provide equations for a mono-exponential decay example, and from that you finally calculate an absolute dose to that volume of interest. The guidelines go through how to calculate uncertainties for each of the steps in this chain, which summarizes into this quite nice and simple single equation. Of course, this guidance has got some limitations. It's only valid for doing mean dosimetry and not voxel-based dosimetry. It uses sphere dose factors, which are then scaled to the corresponding tumor or organ mass, and ignores other potential sources of uncertainty, for example, due to registration between sequential scans. However, this is a very good and easy starting point. It also comes with a fully worked example with patient data, so please use it. And it's also very helpful, of course, to identify where you might be going wrong in your calculations and areas that need improvement. Or the guidance documents to improve measurements of the activity administered into patients, which is the first step in the traceability chain, are available from various places, AAPM, IAEA, ENM, as well as from metrology labs like MPL. Quality control and uncertainty calculations for activity measurements with radionuclides is very important. So again, follow these and get in touch with a metrology lab if you have any questions. And more guidance documents are being currently finalized within the framework of a European project. For example, a good practice guide on how to prepare phantoms with activities traceable to national standards, including recommendations on the options to ensure traceability and how to do uncertainty calculations. Another way that you can improve confidence in the use of dosimetry and improving your own measurements is participating on any type of comparison exercise where you can benchmark your own work against a reference standard or some form of ground truth. For example, in terms of accurately measuring the activity that is administered to the patients, often you will find metrology labs are running national or international comparison exercises to assess hospitals' measurement capabilities and to actually engage with the end users of radionuclide calibrators to identify any needs on the work. Here we have some results from comparison exercises using different radionuclides, mainly therapeutic ones. You can see in light green that in the last few years, 100% of the participants are able to measure lutetium-177 within plus minus 10% of the reference value, and 96% of participants in the UK can also measure lutetium within 5% in the lightest green color, whilst an international comparison found that only 76% were within 5% of the reference for lutetium. When we get to more challenging radionuclides, like for example yttrium-90, which is often used in a microsphere form, things get a bit more complicated. We only see about half of the participants which are able to measure within 5% in the blue colors. If you see a call for participating in such comparisons, I recommend that you get involved. And there's also a Siegfried focus group number 6, which is looking at activity measurements for both yttrium and lutetium to improve traceability. Another way to improve quantification of activity within tumors and organs at risk is to participate in quantitative imaging comparison exercises. This is an example from a European project where we investigated the accuracy, the reproducibility, and uncertainties in lutetium-177 imaging to assess the feasibility of harmonization, which is now currently informing a new EARL SPECT accreditation opening in 2025, which will help towards harmonization of quantitative imaging. And last but not least, you can also participate in dosimetry comparison exercises. This is again a small comparison of dosimetry software where a reference imaging dataset was generated using a 3D printed phantom with multiple compartments that were filled with traceable amounts of activities according to a realistic pharmacokinetic model of lutetium dotatate. The dataset is publicly available in the shown data repository. And these images, together with other calibration data and a standardized reporting template, was provided to 10 participants using various research and commercial dosimetry software options, with the aim of benchmarking each of the steps in the dosimetry workflow against the known ground truth. Here you can see an example of some of the results for the absolute dose comparison, and there is something similar for each of the steps, which allows us to identify potential sources of error at each step and ways to improve each of the steps and minimize uncertainties. More information on the advantages and the disadvantages of the different benchmarking data types that can be used for such initiatives, whether phantom, patient, or simulated data, can be found within Manuel Badí's lectures. So in summary, some key take-home messages. Traceability for nuclear medicine dosimetry is important. Unlike in external beam radiotherapy, it's something that we should aspire to. However, it can be challenging for our field. The measurement, because the measurement chain is quite long and complex, but it's definitely the way forward to improve confidence in the absolute doses reported in clinical studies. However, traceability in itself is not enough. We also need robust quality control management systems so that we can keep accurate documentation of calibrations, uncertainties, etc. Some guidance is available, but more is coming. Please do use it. Uncertainties, again, they are very important and mandatory to ensure measurement traceability. To facilitate this, it would be good if manufacturers included it within the software, so please keep asking for that feature. I will recommend participating in comparison exercises or any other type of benchmarking activities as a way to also improve the accuracy and the reproducibility of your dosimetry calculations. And with that, I thank you for your attention.